Hello everyone, my name is Mathilde Brunel. I'm a PhD student at the Swedish University of Agricultural Sciences in Uppsala in Sweden. My talk today will be about possibility to use porcine, aorta and heart-based product in the diet of patients with metabolic diseases. So this project is about the use of animal products from pork, and more precisely aorta and heart pieces of pork. Animal byproduct can be defined as carcasses, skins, bones, meat trimmings, blood, fatty tissues, horns, feet, hooves, or internal organs. Byproducts from animal origin are rich in lipids, carbohydrates, proteins, and contain bioactive peptides, which can be activated from meat proteins using hydrolysis, cooking, or fermentation. Bioactive peptides are known for their health properties, such as antimicrobial, antioxidative, antithrombotic, antihypertensive. It helps with satiety regulation, as well as cardiovascular, immune, nervous, and digestive systems. Biopeptides can help in the treatment of mental health disease, cancer, diabetes, and obesity. Bioactive peptides are part of a larger group called functional food, and functional food market represents several billions of US dollars since 2015. So what the project is about? It is from a study published by Irina Shenuka. So she and her team investigated the effects of aorta and heart tissues from cattle and pigs on atherosclerosis disorders on rat model. So they had different groups, 5 groups in total. Group 5 was a control group, and 4 other groups which were divided in 2 groups. So the first 2 groups were with cattle meat. The first group was with aorta tissue, and the second group with heart tissue. And they had another 2 groups from pig meat with heart tissue and aorta tissue as well. So the result that they observed was that the treatment rats, which were given heart and aorta tissues, showed a suppression in the body weight gain, an increase in high-density lipoproteins, and a decrease in arteriogenetic index. So the conclusion of the study was that it was suggested the pig and cattle aorta tissues contain bioactive substances effective in the treatment of atherosclerotic disease. And the question today is, can we achieve similar results in human? That's the topic of this project today. So the current hypotheses are that there could be presence of bioactive compounds in the aorta and heart-based product made in the laboratory, and that these bioactive compounds, if they are present, have beneficial effects in blood lipid composition in patients treated with metabolic diseases. To answer these questions, we analyzed the food product made in the lab by Irina Shenuka and her team by analyzing the lipid and metabolite content. We also investigated the effect of this food product on 20 persons during a clinical trial of 1 month by analyzing plasma samples. So about the method for the first part of the study, we had the can made by Irina Shenuka and her team, which is at the bottom left corner of this slide. And she also provided to us the control can, which was bought in the supermarket. And we tried to compare them, and they should have similar composition as they are both made of porcine heart tissue. So the comparison between the products will be made by comparing the fatty acid content of both products and their metabolite content. The fatty acid profiles and metabolite profiles were generated using the following methods. So first, we extracted the fatty acid content of both products by extracting the lipid content of 1 gram of each product using a mix of hexane and isopropanol. We also use gas chromatography technique to identify the different fatty acids for both products. And about the metabolite profile, the metabolite profile for both products were generated by extracting metabolite with a mix of methanol and chloroform and by using a technique called nuclear magnetic resonance, also called NMR. So the result of this first part of the project was about the fatty acids profile for both products. So it showed that test can contain more omega-9 and eicosanoic acid compared to the control. And the control can contain more heptadecanoic acid and linoleic acid compared to the test product. And in general, the test can contain more monounsaturated fatty acid, and the control product contain more polyunsaturated fatty acid. About the metabolic profile part, the test product contain more tyramine, aspartate, O-phosphocholine, and glycine, and the control can contain more sugars, such as glucose, maltose, fructose, and sucrose, as well as more niacinamide than the test food. So in general, we can say that there is a clear difference in metabolite profile between the two products. But the second part of the project, so the method was a clinical study of 20 patients, 10 men and 10 women, who were randomly assigned into two groups, one control group and one treatment group at the beginning of the experiment. The patients were in majority overweight or obese with a median body mass index or BMI of 40 kilograms per square meters. Both groups were assigned a diet of 1,700 kilocalories per day for 30 days, but the treatment group received an extra 100 grams of the test product made in the lab by Irina Shanuka. They received this extra 100 grams every day. And control and treatment group both stayed at the hospital from the first day of the experiment to the 9th day of the experiment. And after the 9th day of the experiment, both groups were told to go home and to continue the low-calorie diet until the 30th day of the experiment. So the treatment group still had to continue the diet with 100 grams of food made in the lab. And during the experiment, the blood was sampled for 3 different time points. Blood was sampled at the beginning of the experiment, the first day at the hospital, also on the 9th day of the experiment, which is the last day at the hospital, and on the 30th or last day of the experiment. The aim of this second part of the project was to compare the metabolic profiles of both groups from the blood sample, and to compare the groups during the 30 days of the diet to see if the test product has an effect on certain types of blood parameters, especially on the blood lipid profile. So to analyze plasma samples, a filtration step was necessary to remove big proteins and viruses which would interfere in the metabolite analysis. So plasma was filtered using special filters made with glycerol to retain proteins bigger than 30 kilodaltons. And then the filtered plasma was mixed with different buffer solution and an internal standard before running NMR sequence. So this is how an NMR machine looks like, if you've never seen it before. And then after the NMR, we get a spectra like this one below, which will be analyzed using different softwares. And so the result for the second part of the study, Irina Shanuka and her team provided data about the body mass index and blood parameters such as triglycerides, also called TJ here. And they also provided information about cholesterol, as well as low density lipoprotein and high density lipoprotein and atherogenic index. So what we observed was the significant weight loss or decrease of body mass index, which was observed between the first day of the hospital and the 9th day for both control and treatment groups, and the weight loss also between the first day and the last day at the hospital, last day of the experiment. So we also observed a decrease in the levels of the different blood parameters, triglycerides, cholesterol, and low density lipoprotein, and atherogenic index as well, but we did not observe any difference in the high density lipoprotein level, and all these differences were only in the treatment group. Regarding metabolites, very few metabolites showed a difference between time points within each treatment. So we actually saw a larger difference between the two groups for all time points. So we had a higher amount of glycine, serine, and succinate in the control group for all time points. And we also saw a higher level of glucose and hydroxy isovalerate 2 and 3 in the treatment group for all time points. So the difference observed in the fatty acid composition were possibly due to different fatty acid profile of the animals in the product. So both products were of porcine origin, but various conditions such as breeding place, feeding type can dramatically change the fatty acid profile. And the large differences observed in the metabolite profiles between test and control can could be explained by difference in product composition. So the control product was not only made of meat, but also contained some vegetables, such as carrots, onions, which could account for the sugar content of the control can. About the clinical part of the study, both control and treatment group lost weight during the hospital stay, which indicates a good adherence to the diets. The difference observed in blood parameters such as LDL, TJ, and CL could indicate possible treatment effects. And very few differences were observed in the metabolites level between time points within each group. One reason for that could be the duration of the diet, which was probably not long enough to see significant results, but it's just extrapolation. So to summarize the project, the two drawbacks of the study were the non-use of the control food at all in the trial, and the fact that the calorie intake was not equal between the two groups, as treatment group had the test product as an extra portion of food, so it was actually not exactly at 1,730 kilocalories. However, the project included a clinical trial, which can be difficult to fund and to get ethical approval. And the clinical study included 50% men and 50% women, which is an important factor to consider when studying the effects of a product on human metabolism, especially with a difference in hormone regulation, which could affect the treatment response. Also, the test product was made in the laboratory by Irina Shanuka and her team, which allowed us to know the exact content of the food, as well as how it was processed. Moreover, during the clinical trial, a hospital stay was planned for both groups, which made it possible to have a strict control of variables and make sure that patients were sticking to their diet. Lastly, we used reliable and sensitive techniques such as DCA and NMR, which gave us results as precise as possible. So to conclude this project, the test product made in the lab by Irina Shanuka and her team could possibly have bioactive compounds, as the test product contained a significant amount of tyramine. Tyramine is a biogenic amine, which is known for its vasoconstriction effect. Moreover, a significant weight loss was observed for both control and treatment groups during the hospital stay, and treatment group showed an improvement in their plasma lipid composition with a decrease of cholesterol, triglycerides, low-density lipoprotein, and arteriogenic index levels. So to find a direct correlation between tyramine intake and decrease in blood lipid levels, more studies need to be conducted. Finally, I would like to thank Irina and her colleague, Elena and Lilia, for their collaboration and for providing food products and plasma samples. Many thanks to my supervisor, JANA, Elisabeth, Galia, Ali, and Sabine for their help in the project. I wish you a nice time during this conference, and bye.